we talked about the structure, right, of the microbe and major parts. Well, a few things worth mentioning before we move on, since we talked about the whole cell shapes. And now look, we're going to talk mostly about bacteria because, let's be honest, most of the pathogens that you're going to deal with in the clinical practice are going to be bacterial pathogens. There are going to be viruses, but you can do almost nothing about most of the viruses. So from the clinical perspective, the approach is tough it out. Bacteria, on the other hand, treatable and cause a huge number of diseases. So when we classify bacteria, well, when we used to classify bacteria when we didn't have any genetic methods, we used the shape. So we've already talked about cocci being round-shaped, right, like Staphylococcus. The bacillus being rod-shaped. Actually, the genus Bacillus, like Bacillus anthracis or Bacillus subtilis, have that shape. Vibrio. It's a rod that is slightly bent, like Vibrio cholerae, the causative agent of cholera. Coccobacilli. I'm trying to remember the pathogen, which is Coccobacillus. It's definitely not Neisseria. I don't remember right off the bat. Bless you. And Spirillum and spirochete are fairly similar. Spirochetes are smaller, thinner, but usually, and the helix is kind of more frequent. Does that make sense? They make more bends. You see, make more bends than spirochete makes more bends than Spirillum. Example of a spirochete, for instance, is a Treponema pallidum, causative agent of syphilis. Okay. Now, we use it for the description because for diagnostic purposes, we're going to look at the culture and we're going to see it. And you look at the culture and you see Bacillus or Vibrio, you already restrict the microbes that the specimen can be. Some of them, mostly bacilli and cocci, can be arranged in a certain higher level forms. What I mean is, remember we looked at the wet mount last Thursday. You picked up your colonies, you put them on a slide. Who picked yellow one? Anyone picked yellow? I think somebody did. You don't remember? It doesn't really, well, the yellow one, the Micrococcus, forms shapes such as diplo and tetrads. Okay? So Micrococcus forms diplococci or tetrads. Staphylococcus epidermidis, forms clusters. And you could see it as well. When you looked at your white colonies, you could see that they are either floating by themselves or form big arrangements, but you wouldn't see anything like that. So if with time, when we know the pattern of organization of those cells, it gives us additional characteristics that helps us to diagnose. Okay, it helps us to characterize, helps us to recognize the pathogen. Bacillus sometimes can form chains. Chains are called streptobacillus. The bacillus that we saw is probably Bacillus cereus. Anyone sampled the big, yes, so Bacillus cereus, it was organized in chains. You could have seen chains of individual cells, right? So you do have to know this, okay, the differences between shapes. I'm not asking about examples yet. We didn't cover that many microbial species. But you have to know what would be a definition of bacillus or a vibrio or a coccus, okay, or a spirochete. That make sense? You do have to know Diplococcus tetrads. What is the difference between these guys? Strepto, and it's a chain of individual cocci, versus Staphylo. There's accumulation. Does that make sense? And Streptobacilli. So know these structures. It's memorization, nothing else. Really, it's just memorization. Now, how prokaryotic cells keep their structure? Because if you think about this, they're tiny, they rather weak, and they are exposed to the pressure of water wherever they live. Pressure actually can be pretty huge. How deep is Mariana Trench in the ocean? Anyone? Any ideas? Huh? 36,000 feet. Pressure at the bottom is quite considerable. Bacteria was isolated from the bottom. There's a bacteria living at this pressure. So they somehow can maintain the structure despite of the pressure. And it turns out it is achieved by osmosis. First, I want to remind you what osmosis is. Yes? Oh, crap. Tell me early, because I forget about this. Yeah, of course I can. So, osmosis. Without looking at the slide, can anyone tell me what it is? Say again? Movement of water through the semi-permeable membrane. Where does water go? In which direction? Huh? High water to low water, right? What in terms of the solute concentration? It's the other way around. Water follows salt. So if you have high concentration of salt, that's where water will go. Does that make sense? There are many ways to conceptualize it. We can go into the concept of concentrations. It's actually illustrated here, okay? So, well, not that well illustrated anyway. Let's put it this way. You have one beaker and another beaker, and they somehow are connected, okay? And you have a lot of molecules of salt, which are black, and a little bit of water. Another one, you have a lot of water and a little bit of salt. Concentration of water is higher in which beaker? In the left, right? So water pretty much diffuses from its high concentration to its lower, sorry, would be better this. High concentration to lower concentration to equalize them. Does that make sense? That tube represents semi-permeable membrane, essentially. So this is semi-permeable. Does that make sense? The whole idea. But we usually say that water goes to where the solute concentration is high. When I just learned it, I made it for myself, the idea that water wants to dilute things all the time. Water wants to dilute things, okay? So if you have diluted solution and concentrated solution, water wants to equalize the concentration. So it wants to dilute the concentrated one. Does that make sense? It's kind of just, water doesn't want to do anything, but it's a neat trick. Okay. So the amount of particles in the solution is called osmolality. This is important. This is number. And it doesn't depend on chemical composition. So let's say you have sodium chloride, salt. When salt is dissolved in water, what happens to the salt molecule? Sodium chloride. Huh? It becomes two ions, sodium and chloride. What about sodium sulfate? How many ions? 3. 2 sodium and 1 sulfate. Does that make sense? Now, if you take same concentrations of sodium chloride and sodium sulfate, which solution will have higher osmolality? Sulfate, right? Because 3 particles. You have 2, this and this, and 3 here. 2 sodiums and 1 sulfate. Does that make sense? It's all about number of ions, molecules, whatever. Okay? That's osmolality. Does that make sense? What I just said. Okay. Now, environment in which you can place the cell can be characterized as isotonic, hypertonic, or hypotonic. So isotonic solution is when osmolality of fluid inside the cell is the same as outside. That's the condition in a human body. Well, generally speaking, in a homeostatic state, it's isotonic, right? So net movement of water is 0. It doesn't go to either compartment overall. That's why we don't get edema or dehydration, something like that. Does that make sense? Okay, good. That's isotonic. When also not concentration, because you can imagine that whatever's in the cell, it's proteins, lipids, sugars, ions, it's all kind of whole bunch of stuff. Okay, so we talk about osmolality. Microbes don't exist in isotonic conditions. They usually actually exist in a hypotonic. Okay. Think about this. If they live, well, most of them exist in a hypotonic, especially environmental microbes. They live in water. They're surrounded by water. And water, just water, like rainwater, pond water, has fairly low osmolality, low concentration of salts, generally speaking. So why hypotonic environment is okay? If environment is hypotonic, so osmolality of the cell is higher than osmolality of the environment, where would water go? Into the cell, right? You can see it right here. It goes into the cell. So what's going to happen to the cell? It's going to swell. And what may eventually happen to the cell? Huh? It will rupture. Well, bacterial cells don't rupture because, well, some of them, most of them, because they have cell wall. Cell wall allows them to keep the shape. They become turgid. Okay? They swollen up, they tight, like inflated, like a balloon. That maintains their structure. Okay? That makes sense. In the isotonic environment, they're going to be okay. They're still fine. They can exist. But in hypertonic solution, water will start leaving the bacterial cell. And do they like it? Cells, what do you think? If they lose water? Biochemical processes are going to get all screwed up in the cell. Pickles. Things like pickled eggs, pickled cucumbers. Why don't they go bad? Yeah, because salt, because it's hypertonic, extremely hypertonic. Jelly. Why? Well, it does go bad, like eventually, But compared to the regular, like take strawberries, blend them with sugar and leave them. They're going to last with sugar for days. Without sugar, they're going to get moldy and, I don't know, very fast. Why? Sugar acts like it creates hypertonic environment, right? So humans learned about preserving foods using either sugar or salt, creating the hypertonic environment, so bacteria are not going to grow there. Does that make sense? So hypertonic is bad. Well, some can live in hypertonic environment. We just talked about Staphylococcus that can survive at 7.5% sodium chloride. And that's not all. Dead Sea. You've heard about Dead Sea in Israel? And you can't possibly, you can't drown. You can die, but you can't drown. Because the salt concentration is so high, it keeps your body afloat. There are microbes that live in the Dead Sea. Okay? The sea salt that is produced in San Francisco Bay is produced by evaporation of water from the seawater. Okay? So they have like giant pools, different salt concentrations. Those pools have different colors, different bacterial populations. Like crazy salty water. Bacteria live there. How can they survive such a hypertonic environment? What do you think they do? Say again? Perfect. They pretty much move the salt into the cell. So for us, it's extremely salty and hypertonic, but for cells that live there, concentration of salt inside the cell is higher than concentration of salt outside. Does that make sense? That's how they maintain it. You put them in the normal water, and they will probably explode right away because no cell wall can hold so much pressure. Does that make sense? Any questions on that? Osmosis. That's how they maintain the shape. Now we're going to chat about cell parts, and then I will give you a quiz. Parts. This are three parts that we're going to start from inside outside. I prefer that way. Sometimes people go from outside to inside. Nucleoid. Prokaryotic cell, the one that we talk about prokaryotes, bacteria and archaea. Okay? Sorry. Prokaryotic cell does not have nucleus, but it does have nucleoid. The dashed line here shows the approximate boundaries of nucleoid. Nucleoid is where the DNA is. You have to understand that DNA in prokaryotic cell is not diffuse. It's kind of concentrated in one place of the cell. Got it? Function is obvious. It stores genetic information and expresses genetic information. And this DNA is not just free-floating DNA. It is bound to proteins. It's kind of like a wire around, like coiled wire, okay? Coiled around the proteins. So when DNA is wrapped around the proteins, genes cannot be expressed. When DNA is unwrapped, genes can be expressed. It becomes accessible. Well, a lot of things, a lot of concepts, you're going to come back to them over and over again, okay? So DNA is bound to the nucleoid-associated proteins. Cytoplasm. It's a solution, it's a gel-like substance. Mostly water, ions, carbs, lipids, proteins, nucleotides, whatever. A part of the cell, fills the cell. Ribosomes. You can't see them on the picture, okay? Like on a photograph. It's a ribosome. Consists of two subunits, small, 30S, and large, 50S. The S is the unit named Svedberg. So the larger the number, like 30s, 50s, it shows how big it is and how fast it precipitates to the bottom of the tube. Because precipitation depends not only on the weight, but also on what? Size, shape, yes, shape. Okay, does that make sense? I mean, if it's large but light, it will precipitate slow anyway, because it's so big. Okay? So these units, the Swedbergs, they are not additive. So 30s plus 50s, when they form a ribosome, that gives 70s, because shape changes. Does that make sense? That makes sense? Okay. Somebody jumps off the roof like this. The person is going to fly relatively fast, right? And then the same person heads up the parachute. Weight is combined, right? And the person is heavy, should fly at the same speed anyway. Parachute opens, weight doesn't change, but shape changes. And the person drops down slow because of air. Does that make sense? Same goes here. Shape changes, rate of precipitation slightly changed. 70s ribosome is unique for prokaryotes. Okay? Ribosomes in eukarya are different. Very different. What is the function of ribosomes? Make the protein. Now, if ribosomes are very structurally different between, say, bacteria and eukarya, how may it help us in a clinical perspective? Huh? Okay. Well, in clinical perspective, it's biotechnology, I understand, yes. But in clinical perspective, are proteins essential for cells? Making proteins is a vital thing. So, from the perspective of treating the disease, and since they are very different, you can shut down bacterial, but not eukaryotic. Does that make sense? You can specifically target the ribosome, and probably 70-75% of all antimicrobials, antibiotics, are focusing on the ribosome because it's so convenient. They're so different. Does that make sense? Inclusions. Not every cell has inclusions. Just a bunch of examples of different inclusions. They accumulate stuff. Can be nutrients. Here is a wonderful, that's just, different how they look under the microscope. They look really awesome. These are inclusions. These are inclusions. You see the dark spots are inclusions. These are inclusions. These so-called magnetosomes. Inclusions that accumulate iron. We have no idea why microbes respond to the magnetic field of Earth, but you pretty much can isolate magnetotactic bacteria that will crawl along the lines of magnetic field. You got to have a magnet and a dirt. They live in the soil. You pretty much shake a magnet and the dirt in the water, then pull out magnet, microbes are going to stick to the magnet. You can put the magnet on the plate and they're going to grow. It's pretty neat. Okay, any questions on these four parts, the inside parts? That pretty much limits what's inside. Okay, that's it. Inclusions, nucleoid, ribosomes, and the cytoplasm. Cell membrane, phospholipid bilayer. So bilayer has hydrophilic. That makes sense? Those are phosphate groups. They are exposed to to the water containing environment. Hydrophobic in the middle. Make sense? That's the lipid parts. So it's a hydrophobic in hydrophilic disguise. Right? Proteins are incorporated in the membrane. Proteins can be transmembrane, like this, or proteins can be peripheral, like this. Peripheral means they are only on one side of the membrane, right? Transmembrane or integral go through the whole thing. They penetrate. Does that make sense? Now, membrane separates the cell and its environment. And the only way a cell can get stuff from the environment is by transporting it through the membrane. The major types of transport. What? Diffusion, right? Here's your diffusion. Diffusion is always along the concentration gradient. Does that make sense? That's your diffusion. Some molecules can diffuse directly through the membrane. Polar or non-polar? Which molecules can diffuse directly across the membrane? Since membrane inside is non-polar. Which molecules? Non-polar, yes. Things like fats, like small organics, like acetone, ethanol. Does that make sense? They can go just directly. Polar molecules have to use some kind of a door, okay? Like a channel. They have to have special structure to diffuse through. They still don't use energy. Remember, diffusion is the movement which does not require energy. Like when you roll something from the top of the slide, okay, you don't have to use energy. This ball here, it will go down by itself. Make sense? What if you need to bring ball back on top of the slide? What if you need to move something against its concentration gradient? You got to use the ATP, right? You got to use energy. This is called active transport. It's kind of a bunch of stuff here. I'm not asking for the details of like how sodium potassium pump works, but basically the idea that I want you to understand that pump moves things against the gradient. Okay, so this slides, quote unquote slides, represent the gradient, the concentrations. So this is the diffusion, movement from high concentration to low concentration. This is active transport, movement from low concentration to high. Does that make sense? You probably know that already. Okay? Time-wise, we're going to stop. Question? Say again? Active transport.